Well, welcome back, everybody. I hope you all had at least some rest over the holiday, if it was a holiday for you. And this is now lecture 10, if I've counted right. We're going to try to finish diffusion-limited growth today and talk about chemotaxis. And I need to remind you that we're being live streamed and recorded. I tried to put some pictures on the cover that reflected what we'll cover today. I have to remember to do that. And I do know that I owe you some graded homeworks, and I apologize for being a little late on that. So today I'd like to get very quick updates on where people are for their projects. And then I want to review what we did last time, which is 2 weeks ago, on secretion and uptake. And then I want to start from the exercise we were doing at the very end of class 2 weeks ago. So that's a homework problem, which may seem so distant that we've all forgotten it. And then we will build on that to do our little simple avascular tumor simulation. And then I want to move into talking about chemotaxis. And the homework that was officially due today was supposed to be exploring chemotaxis on your own to think a little bit about what we need to do for chemotaxis. We'll see how far we get through the exercise. So maybe start out with quick updates. If the answer is you haven't done anything, I understand, but I just want to make sure I hear from everybody before we move on. Joel?
Yes, I finally could get the antimony to start running with the equations. Let me just, I'm running it here. It takes a moment. Let me share. Okay. So here's the equations. I don't know if it's productive to show for everyone the parts of the equations, but basically the antimony, I think we're going over this sometime, if not on this course, in another course with Dr. Glazier. This is basically the functions for the equations for everything that is running on this model. And what I try to do is plot different plots to see by each different molecule if the response is the same as the paper. And I'm having a little bit of trouble of now like making them exactly the same as the paper to correctly say that I'm doing a replication of what is done. And I'm stuck in this place right now, but it's already, I'm glad to see that I am able to plot these things, because it's a little bit complicated when you work with antimony. Volume, for example, is working fine, but some other ones are not so much.
Okay, well, I mean, your strategy of validation is important. Actually, in the Embryo Institute meeting this week, we had Alejandra Magana from Purdue talking about using computing in classes like this one. And one of the things she was talking about was something that we really don't cover much in this class, which we should do more of, which is validation. How do you understand whether a model is meaningful? How do you establish whether it's reliable? And those are fundamental principles of modeling, and they tend to be tacit in the field. In other words, we have an idea of what one should do, but we tend not to cover it because it tends to be something that people think of as maybe not so exciting. And so you want to cover more material rather than that. But then the result is that you never learn it. And I think it is something that would be worth thinking about, especially when you're dealing with very complicated models. The idea of how do you understand whether the model is doing what you expect or not is important. And in the case of the kinds of models in the case of the model you show, that's a deterministic model. So every time you run it, you get the same result. In the case of a simulation like CompuCell, it's a stochastic model. And so every time you run it, you get a different result. And so then if you want to compare your results to something that's published or experimental, you have to ask the question, how do I aggregate my results to know whether or not I actually have reproduced it? Sometimes taking means and standard deviations works, but there are a lot of times where means and standard deviations aren't meaningful, and then you have to think pretty hard about how you're going to do comparisons. So I think those are both interesting things. Great. Connor?
So Andrew and I met to work on the single cell version. And what we ended up doing was that we said, okay, we have a lot of the stuff in place for the single cell. Let's gut everything that is not in their paper and try leaving out the stuff that we didn't feel like was necessary and see if we could tune the parameters just to try out different cell volumes and see if it chemotaxis the right way. And that seemed to work. So now we are officially at the stage where we are looking to move on to having multiple cells. So right now we are looking over how to I think we were looking at I think it's PIF initialization, so we could have randomly scattered cells all over. And once we have that figured out, we'll be able to start including the relative binding. So we'll see what difficulties we run into trying to do that, but so far it seems like we're decently well situated.
I mean, I tend not to do it in this class because it's a little bit of extra work, but it's not that hard to write a short Python routine in your start function that generates cells in a pattern. Now, if you always want the cells to be in the identical initial condition, then creating a file and saving the file which has the cell positions isn't a bad idea. You can also create the Python to generate it and then output it and then read it back in. If you want them to be randomly positioned, then using a Python initializer, you pick a random site, draw a little cell, pick another random site. If it's already inside of a cell, you throw it away, you pick another one until you find an empty spot. Then it's easy enough to say, I want to create 10 cells, 20 cells, 50 cells. You can make them the size and the types you like. And so it would be nice in a way to have something that says create cell of diameter X at location Y instead of having to draw it by hand. But it's not too bad to do that. And you don't really have to make the cells the right size, because the moment you give them a volume constraint, they'll grow within a few Monte Carlo steps to be the right size. So okay. You give the simulation a few Monte Carlo steps to equilibrate before you turn on everything else. All you really need to do is lay down some couple of voxels that are cell A, cut one, so a couple of voxels that are cell 2 in the positions you want. And we tend to do that pretty often. Giuliano, you maybe can give you some guidance on that?
I think Giuliano is already on top of offering guidance, it looks like.
Okay. Yep. Yeah, so here I'm making number to make cells. For each of them, I have a variable that I start as true for occupied. And while the pixel is occupied, I select a random pixel inside of the simulation domain with some leeway on the borders. Then I set occupied to be the Boolean value of the cell field at that point, which is going to be true if there is a cell and false if there isn't a cell. And as soon as this is false, it's going to exit this while loop. And then I actually create a cell here and initialize it at the X and Y, Z that were selected. And here I do have the cell start with some size, but that's because this is 3D and Jim had already wrote part of this. But yeah, the issue is that this doesn't guarantee partial overlap, because if my size here is say 5, I'm only really testing the lower left corner of that cubic initial cell. So I may overlap everywhere else. One thing that I tried to do, and I thought would be neat, and I put it as an issue in GitHub to be incorporated into CompuCell, is instead of doing the cell field of a pixel, do a cell field of a range. So X plus size. And in my mind, that should return a matrix, which is, well, the cell objects that are there or not, which would be neat and fast to do everything at once. But you get a weird error if you try to do that.
Says object is not iterable, probably.
It's even weirder than that. It was, I'll check what happens.
It's okay, but partial overlap is usually all right.
Yep.
It depends a little bit how strict you want to be about having exactly the right number of cells and these kinds of things. If you're willing to tolerate 49 versus 50 cells, and the density of cells that you're drawing is not very high, then the probability of overlap is not very big. If you're filling 2/3 of your lattice with cells, then it's more of an issue.
That's actually exactly what we're doing. It's because it's a 500 by 500 field in the paper with 200 cell population maintained throughout. So I mean, actually that might not be terrible. There's a chance for some overlap, but then if there's the reproduction period, which should reset it to 200, so it should return to that value pretty quickly.
Yep.
I think that you don't have a big problem with this issue.
Yeah, I don't think so. And yeah, you should be able to get away with having single pixel cells to start with, and that let them relax for 20 Monte Carlo cells, steps, and the simulation only begins after that 20.
That would be a good exercise. You could write a little exercise for that.
Yep.
Okay, Connor, was there anything else you wanted to go over today, or was that the...
That covers it.
Okay, great. Josh?
Oh, sorry. No, nothing new to report on my project.
Okay. Alex?
Yeah, I made good progress. I got all the 3D functions converted, but those are really easy to do. So I only have a few more random ones. There's a bunch of like vector and scalar field things. Everything should be done, and maybe I'll look into start working on GPU stuff with TJ.
That would be great. I'm really impressed you're making such progress. We're very grateful to you for that. Andrew, did you want to add anything, or was it covered by what Connor was saying?
Yeah, Connor pretty much covered everything.
Okay, great. Well, I'm around, so if people want to talk about any of these issues, please let me know. Okay, so I want to review a little bit about what we did 2 weeks ago, which is a while ago. And we talked about secretion and absorption. And secretion and absorption are not exactly the same in CompuCell, partly because they don't behave exactly the same way biologically. But you can define secretion in a whole variety of ways, particularly which cells are going to secrete or which cell types are going to secrete, where, with respect to the cell secretion occurs, whether secretion happens everywhere, only on contact with certain other cell types, which is something which is pretty useful. And then whether cells secrete constant flux, that is, a constant rate per unit volume, or impose a constant value at the boundary of the zone of secretion. And that constant value is something that is convenient in a number of ways. It's numerically more stable, and for certain kinds of situations like oxygen in tissues, blood vessels more or less maintain a fixed oxygen tension at their surface. And so that's a pretty reasonable approximation for oxygen or glucose supply. Now, secretion can be defined in CC3DML. It can be defined in Diffusion Solver or Secretion plugin, or we can also define it on a cell-by-cell basis, combining Secretion plugin and Python. And as Giuliano and I checked, if you define secretion in multiple places, it's done multiple times. And the order of execution is the Secretion plugin, then the Diffusion Solver, and then the Python version. And that may or may not be significant to you. And that ability to do that multiple definition may be useful or may be a confusion. But it is there. The reason I mention that specifically is that in some cases, the plugins behave by either overwriting the previous one, canceling the previous one, or having a problem if you have multiple specifications. In this case, they all work together in an additive way. And if we look inside of the CC3DML, the syntax is fairly consistent. In the Diffusion Solver, we have a section called secretion data. And we can either define a uniform secretion rate by cell type, which I've shown here, the first example. Secretion on contact, for example, bacterium secretes, when it's in contact with a wall, a constant rate. Or we could define a constant concentration as a boundary condition. Here, the wall imposes a value of 0.1 at its surface. Using the secretion plugin looks the same, plugin name secretion, and here you have to define what the field name is. Inside the Diffusion Solver, this would be next to would be inside the specification for a particular field, so you would already have specified the field. And then the syntax for the secretion type is identical. So at least there we kept the same syntax for both cases, which is good. And so the cases that we have, we went over in some detail last time. The steps to figure this out, to use Python, would be first we have to define the secretion plugin in the XML, the CC3DML. Secondly, in the step function, we need to get a reference to the chemical field and its secretor. Then we have to say how much to a secretor absorb per voxel per call, because the secretor works both for measurement, actually both, three ways, to measure the field, to secrete into the field, or absorb from the field. Use the same secretor. Specify which cells are going to be doing the secreting, absorbing, or measuring. Determine whether we want it to tell us how much has been secreted or absorbed by that cell, which is usually a pretty useful thing to have. And then only in the case where we secrete inside the cell, we have the option of constant value rather than constant flux. And so we have the following possible boundaries. Inside the cell at the boundary, that would be the one voxel just inside the boundary of the cell. Inside the cell at the boundary on contact, here the red voxels are the boundary voxels that are in contact with the medium. Outside the cell at the boundary, so one voxel outside the cell boundary. Outside the cell at boundary on contact. Inside the cell and at the center of mass of the cell. if you think about it, wouldn't really make sense to have constant value boundary conditions for the top couple of cases, because constant value boundary conditions more or less assume that the cell has a uniform concentration inside of it. It might make sense to be able to do constant value with the center of mass, but I strongly encourage people to avoid using the center of mass option. Historically, that's how people did things, and so if you want to reproduce other people's work, it's not a bad way to do it, but it's a mess, mathematically and numerically a mess to do that. It is faster, a little bit faster execution. And so we can look at the code for this. We need the secretion plugin named Secretion. It can be empty, but you can also put things in it. As I mentioned, they work together with each other rather than replacing each other. Attractor secretor, this is a handle, an arbitrary handle. Self get field secretor, very important function in CompuCell. Then the name of the field, whatever we defined in the PD solver. And then we need to do a call that specifies the secretion. And I've given some examples of this. Secrete inside of cell, secrete inside cell constant concentration, secrete inside cell at boundary with total count. And so you have all those permutations. The secrete inside cell at boundary on contact with. So here the cell only secretes when it's in contact with wall. And you can go down the list and see. I don't have all the permutations here. There are quite a few, I think 14. The one thing to remember is when you use total count, first, there's an inconsistency in the name. It's total count in the function, but it's tot underscore amount in the object that's returned. And secondly, the object that's returned, result, is not the amount, but rather a complex object which includes the amount as an attribute. And so that's something to remember. And the other thing you're going to find is when you do absorption, it'll return a negative number rather than a positive number, so you have to be aware of that. If you don't want to actually absorb or secrete things, there are these convenience functions which operate on the field secretor, attractor.secretor.total field integral. That'll tell you the total amount in the field over the whole simulation, which is a useful thing to do if you're trying to normalize, or just check a sanity check on whether what you're doing makes sense. And then amount seen by cell, which is the sum of the amount in the voxels occupied by the cell, which is also pretty useful. If I want to know how much ATP do I have in my cell, that's the way to figure it out. Okay. Uptake, which we'll work on with today, has a little bit more complexity to it. We can't, in general, take a constant amount out of the field because that runs the risk of making the field concentration go negative. So the default is that we take a fixed fraction of the available amount out with a maximum amount. And one problem that we will run into, which means that we have to display these amounts when we do these, is that these amounts are done on a per pixel basis, and it may not always be obvious what the right values are. The fractional value, take out 1%, 1/10 of a percent, 100th of a percent, makes sense. The saturation value may not be so obvious. And so what you may typically do is look at the amount taken up with the saturation being set to be infinite, and then figure out from the amount taken up where you want to set that saturation value to be. There are a couple of things, one of which I can tag to Giuliano, which is that we really should offer other saturation functions besides the linear saturation. And that would be pretty easy to do because, in fact, CompuCell set up so you can define mathematical functional forms. So that ought to be possible. So that should be added to our punch list. The other one, which is a little bit more complicated and reflects the biology, is that in reality, especially across the cell membrane, the amount of material that's absorbed doesn't depend only on the concentration outside. It depends on the difference in concentration between the inside and the outside. If I have a pipe, the flow through my pipe doesn't depend only on the pressure outside. It depends on the difference of pressure between the outside and the inside. Similarly, when I have flow across the membrane, the flow rate is proportional to the difference in concentration between the inside and the outside. And so to assume that the uptake rate depends only on the concentration outside doesn't really make sense. And so there ought to be a way of doing at least a difference between a value and the amount to give us a flux. The classic linear relationship of that is called Fick's law, F-I-C-K, Fick's law. And we can implement that. That would be a good exercise to do. You can add that, Giuliano, to the stack of exercises. Using that method we did where we did the simulated membrane. But it would be nice to have some of that built into CompuCell. So Joel, what's up?
I don't know if I asked this last class, but is there a way of compensating for the time of exposure of the field in question? Because I don't know if this correctly biologically, but I assume that having the molecule present in the medium for a specific amount of time, the longer it takes, the chances of the cell taking it in is higher than if it's just a burst or just a short period. Is there something like that in CompuCell that takes in account the time?
Well, the rate of uptake should depend only on concentrations. In principle, the rate of uptake could go down if you have saturation. So, for example, if you have biochemical receptors on the surface of the cell, and the molecules binding to those receptors, once they're bound, they can't bind anymore. And so you can run out of receptors. Now, those receptors can then be internalized. The molecule that they're bound to can be stripped off and they can be returned to the cell membrane. Or it can be internalized and destroyed and replaced with new receptors. And so those can give rise to temporal fluctuations. But in general, diffusion uptake rates are really rates in the sense that they are instantaneous. Now, that doesn't mean that you couldn't have some set of equations that describe how those rates change in time. But those typically will be separated from the concentration itself. And so I think maybe more significantly, we're assuming that all these chemicals are moving by diffusion. And that, as we saw a couple of weeks ago, biologically, there's a lot of transport. There's flow. You actually have chemicals packed in vesicles that are carried by molecular motors essentially along microtubules, which are effectively train tracks inside of cells. And so that kind of active transport or movement of a chemical because of a pump, either an ATP-based pump or a symport or an antiport where molecules exchange. You have one molecule cross in one direction, one molecule cross in the other, or two molecules cross in the same direction together. Those kind of pumps can have relatively complicated transport rates as a function of concentration. So The answer as far as CompuCell goes is none of those things are implemented, mainly because there's so many of them. We've seen just in terms of where you secrete and absorb that there are a lot of permutations. And so when you start thinking about pumps and more complex forms of transport, the problem is, where do you stop? In other words, how do you define a set of basic functions that are universal enough that people will use them, but specific enough that you don't have to do everything by hand? And we haven't come up with a good answer for that. If you have ideas for that, maybe it would be worth looking at. Again, the software package vCell from University of Connecticut has a lot of especially membrane-bound transport models. And so in principle, when one sees a package of that kind, and you see a model you like, you can grab the mathematics and implement it in CompuCell. At the moment, you'd have to do that by hand. It's not prepackaged.
The name is vCell, the letter
vCell.
V. Okay, thank you.
Anybody else have any questions about this? I realize it's been 2 weeks, so it's been a while since we looked at that. And here were some examples again of what uptake looks like. Uptake, again, we get the field secretor for the field we're going to be working with. We have inside cell, at boundary, on contact with as options, total count as an option. Again, cell, maximum amount taken up per time step, fractional amount taken up. Here, there isn't an option of constant value. Doesn't make sense to have constant value. Constant value fundamentally can either secrete or absorb, depending on whether the amount outside is less or more. So it has both behaviors. And then you can get the same return of the total amount in that result object. And I'm pretty sure that it returns a minus sign rather than a plus sign for the amount. So you have to change the sign. We'll see that. You can check it today. Okay. So what I'd like to do now is come back to the exercise we were starting at the end of last time, because we're going to work our way up from that. And so to remind you, what we want to do is build a very, very simple model of avascular tumor spheroid. And we're going to walk through the steps of thinking about how to build that. And so this is a little bit of an exercise in how to conceptualize models and build them. And so I want to start out by repeating what was exercise 9.3 from 2 weeks ago. And the idea here is that tumors early on are resource limited. When you have a cluster of cells that are dividing, the cells at the surface of the cluster get plenty of nutrients and oxygen. The cells in the center are starved of both nutrients and oxygen because the cells on the surface are taking up all of the available nutrient and oxygen. In reality, glucose and oxygen are both limiting nutrients. There may be growth factors that are limiting. And the cells are also producing lactic acid, which is fairly toxic, and where the concentration is higher in the center as well. So there's a negative, there's a higher concentration in the middle. There's a lower concentration of things you like in the center of the tumor, and there's a higher concentration of things that hurt the cells in the center of the tumor. We're going to simplify all of that into a single molecular species, which we're going to assume is what you call a critical limiting nutrient. We're going to assume that we can neglect all but one of them. And we're going to ask the question, what is the distribution of that? What are its effects? And typically what you'll see in these tumors is that you'll get 3 layers. At the center, you have a layer, a sphere of dying and dead cells, necrotic core. And that usually represents a substantial fraction of the volume of the tumor. Depending on the tumor type and the size of the tumor, it could be the vast majority of the volume, or it could be some fraction of it. Then there'll be a layer of cells that are not dead, but are sufficiently starved that they're not doing much. They're called quiescent cells. And then a relatively thin layer of proliferating cells at the surface. And the point of this whole series of exercises was to explore the distribution of limiting nutrients in the tumor and how they then affected things like the growth of the tumor and its differentiation. In a slightly more complex exercise, we define 3 cell types, where we define a necrotic cell type, a quiescent cell type, and a proliferating cell type. Here we're going to make do with a single cell type to simplify our life, but changing it over isn't that bad. And we'll define a key limiting nutrient. We call it oxygen glucose. And we're going to, from observation, biologically say that it penetrates a few cell diameters into the tumor. Remember our concept of diffusion length. We want the diffusion length to be a few cell diameters, say 3 cell diameters. And the first thing we're going to do is have a fixed disk of cells. We're going to have those cells take up the nutrient and ask the question, what is the distribution of nutrient like? And it's a little bit different when you go from 2D to 3D because the surface to volume is different, ratio is different in 3D and 2D. And so 3D has slightly different, the ratio of the volumes of the quiescent and necrotic volumes for the same parameters would be slightly different in 3D and 2D. Not dramatically so, but mentioned. So the first thing I'd like people to do, and I'd like people to do this as an exercise now, is to remind yourself, if you have it from last time, great. If not, regenerate it quickly. We're going to draw, and Giuliano, you can do this in real time so that you can show it to them if people are stuck and share the code. We'll use a blob initializer to draw our 3 concentric layers of cells. And looking at this right now, I realize that maybe I should just have stuck with 3 cell types throughout rather than going back to a single cell type in the next exercise, but we'll see. And someone asked about how to do this, and this comes back to the way the stepables are executed, the initialization stepables are executed. And that is in blob initializer, uniform initializer, they're executed sequentially in the order they are in the XML. And so if I draw a circle of cells with blob initializer, and then I draw another circle of cells in the same place, that second draw will overwrite the first draw completely. And so if I want 3 concentric rings like the ones I've shown here, it's pretty easy to do. I draw the big blue disk first, then I draw the green one on top of it, and then I draw the red one on top of that. Now, potentially some of the cells at the boundary, if I haven't been careful, the green cells could partially overlap the blue cells, and the red cells could partially overlap the green cells rather than being nice and clean. In reality here, if I'm freezing the cells, I don't care. And if I were to do this when the cells aren't frozen, the target volumes would relax things so that the pattern would smooth out, which is what we were talking about with Connor a few minutes ago. So please create this. As I say, you can either go back to the code you had from 2 weeks ago or try again. To do that, make the cells frozen, define a constant value boundary condition on the outside of the chemical field, and set the initial concentration of the field equal to that value. Use a single diffusion constant. And I'm suggesting trying a D of 30 and of 10. Assume that there is no uptake in the field outside the cells, and that there is a decay rate here 10 to the minus 3 or 10 to the minus 2 inside the cells. You can do that in the diffusion solver program. And then it's helpful to draw a line across the center of the simulation and plot the chemical concentration as a function of position. So is that exercise clear? I think we did it, again, we did it at the end of last time, but it was a bit of a rush. So I want to give people time to get that working again. If you finish this exercise and you get ahead of people, there's quite a bit you could do in terms of exploring the effects of different values of diffusion constant and decay constant. So or changing the relative size of the domains, or changing the uptake rates for the different cell types. So I want to make sure everybody's on board here. At the moment, I see 2 people who say they're done, and in principle, 3 people who haven't responded. So I wanted to make sure if anybody's having trouble, need help, let me know. Okay. All right. So let's go ahead then and now try to remember what the diffusion lengths are. The diffusion length is the square root of the diffusion constant divided by the decay constant. And so what are, for D equals 30 and gamma is 10 to the minus third, D equals 10 and gamma equals 10 to the minus 2, what are the diffusion lengths for those two? Somebody want to just give me the numbers or not? The answer is there right away.
I mean, to walk through it, you're basically just tacking 2 zeros onto 10, taking the square root, which you're to get a. So that should be, oh gosh, sorry, I'm very sleepy or out of it today.
That's okay. So you have 10 divided by 10 to the minus 2 is 10 to the 3 is 1,000.
Yes. Yeah.
Square root of 1,000 is about 30.
Yeah.
And the size of the cells is on the order of 10. So 30 divided by 10 is about 3 cell diameters, 3 or 4 cell diameters. So the second one gives you a 3 or 4 cell diameter of diffusion length. The first one, on the other hand, 30 times 1,000 is 30,000. The square root of 30,000, I actually can't do that in my head so quickly. The square root of 1,000 is 30. The square root of 30 is 6. So 30 times 6 is 180. So about 200, give or take. And then, so 200 divided by 10 is approximately 20. So the diffusion length here would be about 20 cell diameters. And those are good things to get used to doing roughly in your head. You don't need the exact values, but knowing, is it 1 cell diameter, 10 cell diameters, 100 cell diameters, that's important. So that gives you a sanity check on what you're doing. And then try running it and ask the question, when you use this, do you actually see a diffusion length of 20? And when you run the second one, do you see a diffusion length of 3? What happened? Does anybody have a result they can show?
I finished running mine for the 30, if you'd like me to share screen.
Okay.
Me. Is everyone able to see that? And just so you can see the distribution of the, I think I ended up missizing my cells a little bit because I think that they're a little bit larger. So that might skew things a little bit. I don't know if someone else.
So this is very interesting. Does somebody have an opinion about what this is showing?
I think it's showing that it's not diffusing on the cell, right? It's 0 on the cell.
The concentration is 0 or near 0 in your cells.
Right. I
And
mean,
that-
so. All right.
Go ahead.
Oh, I was going to say, I was just going to try to think out loud as to why that would be. Because I mean, in this case, I don't have it set for being, all the cells are set to have the same rate right now.
So what you would like to do here would be to set the decay constant to be 0
Mhm.
in the medium, global decay constant to 0, and have only a decay constant inside of your 3 cell types.
Okay, so I must have accidentally left one of those.
And here what you're seeing is the concentration goes down dramatically in the outside,
Mhm.
and then is constant inside the cells. And so that suggests that something else has happened. So we could look at your code if you like.
Sure. Let me share screen on that.
Did anybody else get this or not get it? Okay.
So global decay,
0. Okay.
0. And then this is where I tried a few different variations, but this is the one that's active.
Oh, so you've set the diffusion constant to be 0.1 outside. So in line,
Yes.
in line 49. So why don't you make that 30 everywhere? So it's
Oh, for
consistent.
the, okay. Here I can switch back to,
Let's see what happens with that.
alrighty. Okay, that makes much more sense.
All right, so that looks more like what I might have expected. And now if you keep it running, all right, that looks good. Looks like you've reached an equilibrium. And notice that the range of values isn't very large. And you have that inverted U where the decay is a little bit steeper in the middle than on the outside. That's pretty typical. And we could ask a question about what the diffusion range is. And the diffusion range here is large, right? The concentration is actually pretty similar at the surface in the center. Because the difference, remember, you're auto-scaling. So the difference between blue and green is 29.9 to 29.5. So there's almost no difference. So in this case, all the cells get, if this were oxygen, all the cells would be getting oxygen. So now let's try changing those parameter values. Let's try changing the diffusion constant to be smaller. And the alternative one that we said was the diffusion constant of 10 instead of 30. And you have to change it outside as well, probably. But the global diffusion constant up at the top.
Oh, whoops. I just saw a number and there we go.
Okay, so let's try that one now. And so now we expect that the diffusion length will be 3, in which case we should see a big difference between the surface and the bulk. Let's try that. Now the shape doesn't look that different, but you'll notice that there's a difference in 10 now between the, well, maybe not quite 10, 25 to 19, a difference of 5 between the boundary and the surface center. So you've increased that gradient a lot. For the size of the cells we have here, maybe still not enough, maybe not enough, but nevertheless you can change that size. Great. Did other people get something similar? Does anybody need help with that? All right. So the next part of this exercise was the fact that, in fact, not all the cells take up in the same way. In particular, the necrotic cells are dead, so they're not using any oxygen or glucose at all. And the quiescent cells don't take up very much oxygen or glucose. And so the next thing to do would be to change your constants so that there's no decay inside the necrotic or the quiescent cells. And you could run it again. And I'd like everybody to do that, but maybe Connor should demonstrate. Would somebody else like to be the demonstrator this time? Josh or Alex, do you have it? Joel? Again, you're changing just two numbers. You're changing the decay constant inside cells of type necrotic and the decay constant inside cells of type quiescent. Everybody else.
I can do it. If...
Great, thank you.
Just give me a second. This is not the correct one. I have to change it still. Just give me a moment. So I will put for the, I just name it P and Q cell. So for the decay of the P, I'll put like, I'll keep the same. For the Q, I'll put 0, and for the N, I'll use 0 too. Okay. I have a question. I use the camera here to capture, and now it runs every 10 Monte Carlo steps instead of running smoothly over. Do you know how I cancel that?
I think, actually, I don't know that. Giuliano, do you know?
So it was updating every Monte Carlo step, and you've asked for a screenshot, and now it's only a word.
Yeah, because my screenshot is every 10 Monte Carlo steps to take the images for the thing. And I did this the last time we were doing it, and now I can't cancel it back. I think I have to change. I think I put 1 through 0. Yeah, and I probably should use the 30 instead. Just give me a second. So it'll be the same as Connor.
That's right. You have to remember you have that background diffusion specification. There you go.
It's weird because I think it's I think it still takes in there. So the decay should be 0, right, for Q and P. But the diffusion coefficient should also be 0, right?
No, the diffusion coefficient is the same.
Oh, okay.
Because the diffusion coefficient is saying is basically that for water.
Mm-hmm. It goes through the same way. Okay.
Yeah. It might actually be slightly higher in the necrotic region, but not by a lot.
Okay. Yeah, I got this weird shape.
I think that's because your cells are not centered on the lattice.
Mm-hmm. Yep, it's a little bit offset.
Yeah, yeah. And if you're cutting down across the middle, then the symmetry isn't right.
The middle is about about here.
Yeah, just slightly off.
Mm-hmm. Has more cells of this type here, and then
Okay. So if you don't have the uptake in those two central regions, then of course what you'll see is something flat because the concentration will be more or less uniform. There isn't any place for it to go.
Mm-hmm.
In this case, the values are almost all the effective uptake is very, very small, which is a little bit surprising to me. Is this for the long diffusion length and the small decay constant?
Yeah,
This is for 30. This is for 30 and 10 to the minus 3.
Yes.
So why don't you change it and do the 20 and 10 to the minus 2?
Okay.
See what that looks like. And that one you could actually do by hand. You could go into the model editor on the left and do it there too.
It's true.
Is there an issue with doing this type of simulation in 2D in the sense that there's resources towards the middle that can be pulled from? Or I get, oh, actually, no, maybe not. I was thinking about it as like secreting constantly from the background, but that's not what this does. I answered my own question. Never mind.
Where there is a difference is the following. If I draw the circles with given radii, so I say the red region is the blue region is between radius 0 and 3. The green region is between 3 and 5. And the red region is between 5 and 7, say. And I ask what fraction of the cells are blue, green, and red. That's different in 2D from 3D. Because in 2D, I have a circle and a disk, so I have area is pi r squared. And it's pi r squared outer minus pi r squared inner for each one. If I'm in 3D, it's pi r cubed, 4/3 pi r cubed. And so the volume goes like the cube of the radius in 3D and the square of the radius in 2D. And so what I'll find is that the blue region will be smaller relative to the green and the red in 3D than it would be in 2D for the same radii. That's what I meant by saying it's a little bit inconsistent between the two. So, okay, good. Where's my slide? And then maybe we don't need to do it, but you could try this at home. It was in the homework exercise. If the quiescent cells take up an intermediate level, then you'll get something, again, intermediate between the two cases that we've seen. That smooth curve, which looks like a parabola in one case, and the flat bottom, you get two slopes. And so here were just some results that I did a while ago. In the case for a long diffusion length and the case with a short diffusion length. This was the short diffusion length one, I believe. As you can see, there's a big difference in concentration through the tissue. So now let's think about trying to do this in the context of a growth. And I've already said what's on this slide to some extent. In reality, cells need quite a bit of oxygen when they're going to divide, because they're doing a lot of protein synthesis. And they certainly need glucose, because glucose both supplies energy and the building blocks for cell growth. If metabolic resources are limited, you're hypoxic, or you don't have enough glucose, the cell basically goes into a safety mode where it will survive but not grow. And if the metabolic needs of the cell to survive aren't met, then the cell will die. And typically, that kind of cell death, when you really are starved, is a necrotic cell death rather than a programmed cell death. And so the cells tend to fall apart and die in a disorganized way. Sometimes they die in an organized way. And so here is a picture showing essentially the proliferation zone on the outside, a quiescent zone inside of that, and then it's hard to see, but there's a small necrotic core in the middle. And this is typical of almost any solid tumor that doesn't have blood vessels growing in it. Now, depending on what rules you use to determine the thresholds for cells proliferating, becoming quiescent, and dying, you can change the widths of these domains. And those widths can be very sensitive to the diffusion constants and uptake constants you've chosen, or they can be relatively robust to them. And there's a fair amount of subtlety there in terms of defining that. The other thing that you'll find that is a subtlety in this kind of modeling is that it matters where the nutrient is provided. If the nutrient is coming from the boundaries of the simulation, then what you'll typically find is that the amount of nutrient available to the cells goes up as the cells get closer to the boundary, because you don't have to have the nutrient diffuse across the empty space. And so you get something rather paradoxical, where actually the growth rate will go up as the cells almost fill the container. Alternatively, and this is true biologically, especially for oxygen, and in tissues that have a lot of blood vessels, you could imagine that the medium itself is providing the nutrient. And in that case, the total amount of nutrient being provided goes down as the tumor grows, and there's less medium to provide nutrient. And in that case, you get something that's more self-limiting, where the tumor will grow until the amount of resources the tumor is using balance the amount of supply from the outside. And so it's a little bit easier to do, to tune your parameter simulation when the nutrient is coming from the medium than when it's coming from the outside edge of the simulation. They do give slightly different results, and it matters. And the biology will determine ultimately which one you have to do. So if we think about cell growth, the first thing that we should be aware of is that the cells actually have what's called resting metabolism. They need a certain amount of nutrient and oxygen just to survive. And they can't grow. If they have an amount above that, they can grow. And the way I've got this written here isn't really very pretty, and I should probably redo this slide a little bit. But we could imagine, to begin with, that the target volume of the cell grows at a constant rate. That's what the first one is trying to say. And In the second case, if we say that S is the amount of surplus nutrient available to the cell, the uptake amount minus the resting metabolism, then we could say that the target rate, the target growth, the target volume is the target volume plus some growth rate times, here we're assuming the cell doesn't shrink, the maximum of the available nutrient in zero. So if the cell doesn't have enough nutrient, it stops growing. Now, you could imagine that it shrinks if it doesn't have enough nutrient, in which case you wouldn't have the max function. And you could include something like pressure-limited growth, which we did a couple of weeks ago, in which case your growth rate would be a product of the pressure, limited growth, and the maximum and the uptake. So if we're going to do any of these things, we need to use that total count because we want to know how much of the nutrient we have available to us. And so I want to remind you of the syntax here. Instead of using the decay that we just did in the example we worked through, we'll turn off that decay rate inside the cells. We'll define a secretor, and then we'll actually have the cells individually take up nutrient and report to us how much they've taken up. Is that clear? Very good. And so let's do that problem together. So what we want to do is model the interaction between nutrient availability, growth, and the transition from proliferating to quiescent necrotic cells. We won't do that transition very much here, although the way you've got it set up actually in the simulation you're working from would let you do that if you unfreeze your cells. And the actual rules that govern that transition between proliferation, quiescence, and necrosis are pretty complicated. It's not just a single level of a single molecule. But for the moment, we're going to assume that that's the case. And we'll do this in steps. The first thing we'll do is we'll do the uptake, and we'll look at how much nutrient each cell gets. And then we'll say, okay, we're now going to define a threshold, which is going to give us a band of cells at the surface that are going to divide, grow and divide. And then if we like, we can define a set of switches that give us quiescence and necrosis. And we're going to use this trick that I just described. We'll assume that the surrounding tissue around the tumor has got plenty of blood vessels, and so it has enough oxygen, glucose, and we'll have the medium then secrete at constant value. And so the exercise is the following. Let's create a simulation, 100 by 100 lattice. You don't have to get rid of your, I wouldn't get rid of your necrotic and quiescent cell types. You can use the simulation you've got now. You will need to unfreeze the cell types. And here I said call it cell, but just use your proliferating cell type from before. You have your chemical field, whatever you called it, diffusion solver FE. And now you want to turn on cell growth, division, contact, and volume local flex in your simulation. And then we're going to set the global diffusion constant. Here I've said 0.1 instead of 20 in the other one. We won't have any global decay. And we can set the constant concentration of medium cells to be 1. We can scale it to be any number that we choose, as long as we keep consistent. Start with a single cell in the middle of the simulation, and I set the maximum growth rate to be 25 over 500, so the cells would double in volume every 500 Monte Carlo steps. To begin with, we're not going to have cell division. And then use the secretor to take up chemical. Here I said the maximum uptake was 0.05, relative uptake was 2.5%, sorry, 0.25%. Remember, this is each time step, so you're taking this up frequently, so that's why these numbers are fairly small. Add a tracking field to display the amount taken up by each cell. If you want to use that midline plot that you've been doing, that's great. And then run the simulations. And then why don't you see whether you can determine what's the maximum uptake for the particular case you've defined inside of any cell, because you'll need that number to scale everything going forward. So is that exercise reasonably clear? Joel, what's up?
Yeah, I think I have that simulation. I think ready here. Can I show you? Can I show everybody? And okay. And then we can.
If everybody's already done it, then you should tell me, and I can skip on to the next thing. If people,
Yes.
yeah, go ahead.
I was just going to say I have something similar to this. I think Joel has a couple of additional plots that I don't have, but I have a simulation that is something like this.
Yeah, what I did here is instead of like making the cells change to different cells, I just plot the cell number, accounting for them as changing. It's because it was like late, so I didn't have the time to go over changing everything. But as the simulation goes, you start to see the depletion of material in the center, and start to see the cells in the middle will grow slower. It's basically becoming a factor of growth. I don't know if the zoom is covering something here, but yeah, just let me know.
That looks fine. Let's let it run a little bit.
Yep.
I
Yeah,
like the way you did that plot on the right. So the plot on the right is telling you that the cells in the middle are having very low uptake, and the cells at the surface are having a higher uptake.
Yeah, we can see that this is basically the boundaries, or the cells in red, the mean intake and the blue cells.
So maybe the max growth rate that I defined was too slow, and we should have used a slightly faster growth rate for this, so that we don't have to wait quite so long for the simulation to run. But what you got is fine. Let's keep taking a look.
Yeah, the
And
number
so,
of cells there are necrosis is going high. I don't know why. I think it's changed so fast from one to another that this is not stable or something.
what kind of rule did you use to determine switching from proliferating to quiescent to necrotic?
It was completely arbitrary. I think I just visualized how much the intake in the mean or the max intake was, and then I consider, I think, 0.4 as my guide for a Q cell, and less than that, I would consider it an N cell. So if it stays on the, they have a short band. If they stay inside that band, they still Q cells, but then quickly they become N cells.
So you have two thresholds or one threshold?
Yeah, two, because one is for becoming Q and the other one is for becoming N.
Can your Q cells turn back into proliferating cells, or is it irreversible?
That's a good question. I don't think so, because I don't have a test to see if any Q will uptake more oxygen in this case. But I think it's something easy to change if we want to keep pushing more oxygen, because now it's constant everywhere.
So in this case, the uptake is quite efficient. Basically, only the surface layer is getting anything.
Yeah, only the very edge. And they stay the same size too. We start to see that they don't grow as much as the other ones in comparison here. Although they don't freeze or they don't die in the sense of like being stable, they keep moving. That's something that I could also do. But one interesting thing is soon we start to see that like half of them are basically dead compared to this is the total amount number, I think, because I didn't change that. But half of them are basically dead. They're the half on the center.
So one way to change this, I mean, if you change the fractional uptake, the problem with that is it'll change the amounts, and therefore you'd have to change the thresholds too. Unless are the thresholds defined relative to the maximum, or are they defined as absolute numbers?
Yeah, absolute numbers. Yes, it was.
Yeah, so then you'd have to change that. If you change the diffusion constant, if you made the diffusion constant higher, then you should see more proliferating cells, or at least the surface layer should be thicker. So that's something you could do actually while you're running. If you go into the diffusion steppable, that's and then change the diffusion constant.
Change it for a higher number.
So I see. I have a question.
Yes.
What's the diffusion constant in the cells?
Oh, that's a good question. Let me see the code here. Just give me a second.
I think maybe that's why you have such a certain, so the diffusion length is so short.
Yeah, it's not defined.
So it's 0 in that case.
Yeah, it's 0.
So this is why you're having only the surface is getting nutrient. So why don't you try running it again, putting a diffusion constant in. You could start with a global diffusion constant. You could just do that by hand here. Make the global diffusion 0.1. See what happens.
I hope it doesn't crash. Oh, I can't because it's int, I suppose. It's 1 now. Okay, yeah, now you can start to see that the end cells got back to, yeah, they're all growing. So it's reversible, basically.
So the state, it reads the field and adjusts the state dynamically. Okay, that makes sense. So now the number of, let's see, so basically now it looks like we've got a situation where the diffusion constant is so big that at the constant that everything is turned on.
Yeah, everything is taking a lot of you can start to see that even the mean is above the threshold that I used.
But now it's getting big enough that you're getting maybe a few cells in the middle that are not fully oxygenated.
Yeah, but even the blue here, it's 0.6. I think I put 0.4. I can change that too and run it again.
Well, let's see. The medium is supplying, right?
Yeah.
Okay, so if this grows, and what would happen if it filled the entire lattice?
I don't know. I never run above, I don't know, it's almost 7,000 now. I never run completely.
Does anybody want to predict what will happen?
I mean, effectively, shouldn't it respond as though every cell is in the middle, right? Because they're all surrounded.
Well, but especially, what happens to the amount of nutrient available?
Oh, it's going to plummet.
Right. So if you fill the whole lattice, there is no nutrient. So all the cells they become necrotic.
But when it comes to global diffusion in this sense, in this case, isn't it is just being supplied from the medium, because I use secretion from the medium as a
Right.
way to
So
supply.
if there's no medium, there's no supply.
Okay. Even with a global diffusion. Okay.
The diffusion tells you how fast things move, but they don't. They don't create things to move.
Got it.
Okay, wait. So that means that once this fills up, there's not going to be anything else being supplied by the medium. So I should think about it as like overtaking the medium as opposed to like laying on top of it, right? Okay.
This is imagining something like a section through a 3D object. And look what's happened to your glucose uptake now.
Yeah, everything is going down here. We start we'll start to see this cell going up too. I hope.
Now what's not going to happen here is you don't have any way of having the cells die and disappear, so you're not going to get medium back. But what would tend to happen in this sort of situation is that you get oscillations where the cells would overgrow, then they die back, and then they'd overgrow, and then they die back. And that does happen with real tumors. In this case, you don't have that mechanism implemented. And you'll notice that if you look at your concentration field, the maximum concentration keeps coming down because all you have is what's left from the previous secretion. If you look in the top right image.
The top right here?
No, top right of your picture.
Okay.
Max is.
Yeah, here is also. But it's interesting, it doesn't react the way I was expecting. I was expecting this to. It's trying or something.
Yeah, I'm not sure. Looks like you're getting some quiescent cells now.
Yep.
Because again, it'll depend sensitively on your rule.
Yeah.
For what the transitions are.
Yeah, now they start to transition from one to the other.
Yeah, level-based transitions tend to be finicky.
And it was completely took out of my head. I didn't use anything dynamically in the sense of getting a reference from the cell as a way.
Yeah.
Yeah.
Does anybody else have something they want to show? That's a good exploration of this problem. Good is maybe not useful. It's an interesting exploration of this problem. It's a big problem. In principle, you could have. I mean, there are quite a few things one can do with it. So you've done a bunch of. I mean, you've jumped ahead. I mean, you've done quite a bit of this. My next assignment was going to be doing this, which was to change the growth rate as a function of the uptake amount. And this was an example that I did, just creating the diffusion field. In this case, I had medium secreted a value of 1, global diffusion constant of 0.1. And then I have the cells secrete, uptake, 0.05, 0.0025. I really should have had total count here, not just uptake. And in this case, for the particular values I defined, there was a big difference in concentration across the cell, and the penetration depth of the field was about 2 cell diameters, 2.5 cell diameters for these particular parameters. And again, I had to do absolute value because you get a minus sign returned by total amount. Created a tracking field, which you already did. And here was my version of your code. And you can see then that the maximum uptake in this one was 0.08. Min is effectively 0. And so that means you could say, if you have a threshold of something like 0.075, you'll get proliferation just at the boundary and not in the center. So I did that, had the growth rate depend on the uptake amount. Mine also is not centered on the lattice. That doesn't matter so much if you have the medium secreting. If you have the boundaries secreting, then it's more significant. And one can play with this. Just as a sanity check for people, maybe Josh. One of these two has a higher diffusion constant than the other. The upper row or the lower row? Which has a higher diffusion constant?
So if I remember, the diffusion constant is inversely proportional to the time it takes to diffuse, correct?
Right. And the diffusion length goes which way? Higher diffusion constant means bigger diffusion length or shorter?
The diffusion length is proportional to root d over the time constant.
Yeah.
So bigger d means a longer length.
Right. So which of these two, the top or the bottom, has a larger diffusion constant?
So 1 over a big number is a small number. So
Well, if I look at the picture in the middle, say, the chemical is getting in further in which one?
The top row.
The top one. So that means its diffusion length is larger. That means the diffusion constant is bigger on the top one than the bottom.
Yeah.
So again, it's not the reason these are sort of simple thought exercises because it takes time. You have to practice to get used to these things. Notice in the one on the top, there are a couple of cell layers that get a reasonable amount of chemical. The bottom one, it's more like Joel's original simulation, only the surface ones get anything. And so if I had proliferation that depended on the uptake, this lower one, only the surface cells would proliferate. And this one, a couple of layers would. So the next thing would be to add basal metabolism, which is to say then that the growth rate isn't proportional to the uptake, but it's proportional to the uptake minus a metabolic rate. And then we could do a variety of things we could do. We could say the cells don't grow if the amount of taken up is too small. Or we could say actually that they shrink if the amount of uptake is too small. So why don't people try this little exercise for a few minutes? You can maybe one or two of you can try, I don't know, Joel and Josh, why don't you try the first part of it, which is to say that the growth rate is either 0 or the absolute value of the uptaken amount minus a basal metabolism. And two of you, say Connor and Alex, try having the growth rate be able to be negative. So it's just the amount taken up minus the basal metabolism. And let's compare that. Try that. Is that okay? So why don't people give that a few minutes to try that? I'll redo the poll, but don't answer the poll until you're ready, basically. So I can just keep track of things. And give people a minute to give that a shot. If you've got the base exercise done, there's, as I say, you can play around with the changing the effect of changing the diffusion constant, changing the uptake rate, changing the target, the metabolic rate. Why don't I walk through some material for just thinking about this, and then we can have people show their results. So here were some of my results. Depending on the parameters I chose, I could either get something that was relatively, if the diffusion constants and the uptake were both pretty high, I got something that looked relatively round. If I made the diffusion constant smaller, I got something that looked spiked. And that's typical of what's called diffusion-limited growth. In this case, in the top one, I don't have cell death. Cells stop growing, but they don't have death. In the bottom, you can see in the middle, they're shrinking cells. And so I have contraction in the middle with expansion at the surface at the same time. And I don't know if people who tried, I don't know if the people who got it done, if anybody got it done with one case, the case where there was no shrinkage and the case where it was shrinkage. But I wonder if you got something that's similar. Giuliano, what about you?
Working on it, it's, as you said, fiddly.
So, we're a little bit behind in terms of time. Why don't we take a break? And if people would like to continue to work on this exercise during the break, that's fine. And if there are any questions about it, we can come, when we come back from the break, we can answer questions. If not, this was a homework problem, and you can work on it, continue to work on it as a homework problem. I do want to get to chemotaxis, and so I'd like to move on from this to chemotaxis. There's a lot one can explore in this problem, so I don't want to shortchange it, but I also want to make sure we get some chemotaxis work done today. So let's meet back here at 7:05. Did it work? Did you get something similar, something different?
Yeah, mine was a little bit different than yours. I know you told me to do the other one. I did. It was similar to what I already had. That is the top one. Then I tried the new one where the cells disappeared. But I think the rate of growth of the cells it's equal to the rate of death. So mine is a little bit more roundish than yours is looking.
You want to show?
Sure, it's running. Yeah. They get smaller and smaller. But I think they divide at the same rate as the other ones are dying, so that's why we're seeing not much.
Well, that's actually a success in the sense that in reality, that's more or less what happens. Avascular tumors reach a steady state, well, it's equilibrium, where the number of cells dying and disappearing is comparable to the number of cells being generated. And so you get these avascular tumor spheroids will grow. They'll get to be typically 1 millimeter or 2 in diameter. Some tissues they can get be bigger than that. And then they'll be relatively stable.
Yeah, the interesting part that I think was this, the waves you were mentioning before. I got the same thing here. And the uptake is also doing the same thing, even on the min, but it's hard to see because it's very small.
Yeah. Did anybody else get something that's comparable to that, or?
I think that there's a bug somewhere in mine, and I'm trying to figure out if it's something that was wrong in code that I thought was previously working, or if it's something I just did. So I'm making some plots right now to try to figure it out.
Anybody else? Josh, were you able to get any of that to work?
No, I was having trouble with it.
You need help, or would?
I mean, I'm still in the process of debugging.
Do people want to spend more time on this, or do we want to move on? I'm okay either way. If you want to take some more time, that's fine. It's no skin off my nose to wait another few minutes. If you want to work on it some more.
I would actually vote to move on because I have a question on chemotaxis that I want to show you, like some behaviors that I got on the exercise. But it's one vote. Other people had their votes too.
Connor and Josh, do you want to keep working for a few more minutes, or?
I also think it would be beneficial to talk about chemotaxis,
Okay.
and I think that with the Discord, since Joel's code was working, I could probably just poke the community as I try to figure out what's going wrong with this exercise.
Okay. Okay. Well, then we'll move on, and if people want help with this, we can certainly come back to it and work on it together. So I'd like to now talk about chemotaxis, or more generally, cell movement and gradients. And we've already seen at some level gradients in the sense that we had a gradient of concentration that led to a change in the cell growth rate. And one of the things that will happen if you have cells grow and divide in response to chemical and die, if they don't have enough of it, is that in the example we gave where there was a uniform source, you got a circular tumor. If you had a source that was localized in space, imagine a blood vessel here that's a source of a chemical, then the cells that are on the side of the tumor pointing towards the blood vessel will grow fast. The cells that are on the far side are shadowed and will die. And the tumor, even though the individual cells aren't migrating, the tumor as a whole will migrate towards the blood vessel because it will grow on the side of the blood vessel and die on the far side, and it will gradually translate over. And that happens. I mean, you can think about this also with something like plants, which don't move once they've started, where you have a forest with a water supply that changes. It's near a water supply, and the river moves, and the cells the trees that are far from the river now die, and you get new trees growing which are near the river. And so, even though the individual cells aren't motile, the fact that they're growing or dying selectively can lead to movement. And we'll actually come back to something that's a little bit similar to that in a minute. So one of the main ways that cells do respond to gradients is by moving. And in particular, cells, we keep talking about chemotaxis, and we'll define that in a second. But cells can alter their movement in response to all sorts of things. They can move in response to the stiffness of the substrate. Cells often prefer to move on rigid materials than soft materials. And that has to do with the way cells move. Cells create bonds to their substrate and pull on them. And if the substrate's too soft, they can't get a grip, essentially. Cells can move in response to electrical fields, especially in wound healing. Cell migration is often driven by gradients of electrical potential. Certainly, cells can move in response to gravity, both because of the direct gravitational force, but there are also cell types that can actually respond, measure gravity, and directly respond to the gravity that way. Some kinds of frog embryos have special particles in the egg that measure essentially the direction of gravity. I saw them. The front page of this month's Scientific American was about magnetic navigation by birds. It's not quite the same scale, but also responding there to the magnetic fields. I don't think any cell level behaviors typically respond to magnetic fields. They're too weak. It's not impossible to think about. Cells definitely can move in response to temperature. They can either move up or down temperature gradients depending on what their ranges are. Some cells respond to light, either moving towards or away from sources of light. They can respond to acidity, pH. There definitely are cell types that respond to fluid flow. That can be either in terms of movement or in terms of proliferation. The cells, the endothelial cells lining your blood vessels, tend to proliferate if there's a lot of flow, and to degenerate and die if there's not a lot of flow. Many cell types respond to being stretched or periodically stretched and compressed. If that weren't the case, working out wouldn't work. But your bone also remodels in response to periodic stresses. If there's insufficient stress, your bone gets thin, degenerates. Of course, excessive stress can break bone. And so there's a rather complicated relationship. It's not just the total amount of force, but also the temporal pattern of force is important. And then classically, chemical concentration is an important thing. That chemical concentration can be diffusing, and we typically think of chemicals diffusing in the extracellular environment. But we'll do an exercise, I hope, before the end of the day where we have non-diffusing chemicals. And the two main ways that cells move are chemotaxis and chemokinesis. And again, we could have things that cells like nutrients or signaling molecules, and things that cells tend to avoid, like toxins. Sorry about that. So it's possible for cells to move generically up towards higher concentrations of whatever it is, down towards lower concentrations, or sometimes they have some sweet spot in the middle that they like. And whether they respond in a linear fashion or nonlinear fashion, uniformly or with temporal pattern, varies a lot. The two classical responses to chemical fields are chemokinesis and chemotaxis. Both of them have the same net effect, but very, very different mechanisms at a microscopic level. Chemokinesis is what bacteria do, and the theory behind chemokinesis, which goes back to Purcell in the 1950s, is that bacteria are very small. And because they're small, they can't measure a spatial gradient across their volume. The concentration at one end of a bacterium and the concentration of another at the other end of the bacterium are almost the same because the fluctuations in concentration are large compared to the gradient, the difference in concentration across that small size. Bacteria typically half a micron across. And so to measure spatial concentration differences, what they do is they integrate over time. So they turn spatial concentration differences into temporal concentration differences. And we'll say this again, but in this case, what happens is bacteria move in a straight line, and they ask the question for themselves, is the concentration of the thing that I'm trying to seek going up or down? If it's going up, they continue to move in that direction. If it's going down, they stop moving. They don't have the ability to orient, so they pick a random direction, and they do it again. And those straight line movements, or quasi-straight line movements, are called runs, and the reorientation is called a tumble. And it may not be immediately obvious that random movement can actually get you up a gradient, but it can. Again, because you change the time you spend in your run, depending on whether the situation is getting favorable or not. And the bacteria have rather complicated set of signal integrators that determine the duration of runs versus tumbles. These are some pictures from that beautiful Physical Biology of the Cell textbook showing how bacteria actually swim. They have flagellar motors. If they're spinning counterclockwise, all of the flagella match up into a single corkscrew, and the bacterium swims forward. And they can move pretty fast. They could move 10 to 30 microns a second, which is quite a lot, given that their size is about half a micron. So they can move 20 body lengths in a second, which is quite fast. If the motors start rotating the other way, then all of those individual corkscrews spread out. You see that here in the tumble picture. And the bacteria then gradually diffuse in direction, randomize their orientation. Then they reassemble their flagella, start spinning counterclockwise again, run, and tumble like that. If there were no bias to the duration of the runs, then the bacteria would diffuse. It'd move randomly like a molecule in water or gas. However, if runs that are going increasing the value that I care about are longer than ones decreasing, the net effect is to move up the gradient. And so even though I'm zigzagging and sometimes moving in the wrong direction, the net effect is to move up the gradient. And that's a very powerful way of exploring space. Eukaryotic motility, the cells are larger, and so they can actually measure concentration differences between one end of the cell and the other. And so in that situation, the cells actually are able to swim actively. In this case, part of the cell is moving rather than the whole cell. And you see these other cells swimming around the amoeba here. But the cell could actually actively protrude its membrane. And to do that, it typically uses cytoskeletal machinery, in particular actin cytoskeleton, to do protrusion. Now, while we typically think of that kind of movement as protrusive, in fact, the main forces that act during eukaryotic cell movement, cell crawling, are contractile forces. It's not so much that the forward edge is being pushed forward, it's that the backward edge is being pulled back strongly. And since the volume of the cell is conserved, the net effect is forward movement. There are a number of different kinds of forward movement. Amoeboid motion, which is being shown here, leading edge formation, which is typical of cells crawling on flat substrates. Keratinocytes are the classic example of that. And filopodial extensions, where the cell will protrude narrow fibrous projections that anchor onto a target and then pull the cell and contract and pull the cell along with them. So you can think of throwing out a tether and then pulling yourself up along the tether. Bacteria also have some movement mechanisms of that kind. Beyond the swimming mechanism that I showed, there are pili in bacteria that can do rather complicated things. Some of them work like little jet engines, pushing material out of the cell and pushing the cell forward with it. Others actually do the same thing, where they'll tether and pull the cell along. But in general, there's a lot more tension-directed movement than there is extensional movement in cells. Just the way our muscles contract, they don't push. And so a lot of our of the way our bones and ligaments work is trying to convert contractile motion into forward protrusion. And so in our modeling, we're going to assume that there's a forward force on cell boundaries. But we need to remember that at least at some level, that's a bit of an oversimplification. One of the classic places that you see this kind of movement is in dictyostelium. There's this famous, I guess I can do without the background music. I didn't know that that had background music. This is an aggregate of cells called the grex, or a slug. These are individual slime molds moving one at a time. And of course, one of you has decided to work on a problem not with cellular slime molds, not with dictyostelium, but rather with true slime molds, which don't have the cellular organization. But here you see cells individually and as clusters moving towards a source of a chemical, in this case cyclic AMP. Now, the cells themselves are secreting that chemical as they move. And here's an example where there's a pipette, a micropipette with cyclic AMP in it, and somebody is torturing these little cells by moving the target around so that the cells chase the target, and then they'll move it. Now, maybe this one they don't move it. Sorry about that. There's another one where they periodically move the target and the cells chase it, and then they move it and they chase it and they move it. But you see here clearly almost a rather simple forward-directed movement. The velocity seems to be relatively uniform, and they seem to move up the gradient pretty directly. It's not random movement. In reality, there's some pulsatility in that movement. It's not simply forward at a constant velocity. And the dicty have this cyclical movement that's rather special. And this is their behavior at a large scale. So you saw individuals here, small clusters here, and this is visualized on a much larger scale, and you see these complex pattern behaviors. Due to those are density fluctuations in the bacteria, in the amoeba. So we're going to assume, to begin with, that the chemical diffuses everywhere and that the cells don't affect the field. In 2D, that's not unreasonable if you think of the cells crawling on top of something. In 3D, it can't be right because the cells are going to exclude the chemical that they're moving through. But as an first approximation, it's not too bad. And the cells the chemical doesn't actually exert a force on the cell. The chemical causes the cell to change its behavior in a way that causes the cell to move. And so the cell is the thing that's creating the force. The chemical doesn't create the force, it sends a signal promoting force creation. However, in many cases, we find that the velocity of the cell is proportional to the gradient, in which case, assuming that the force is proportional to the gradient of the chemical isn't a bad approximation. And so the simplest assumption we're going to make is that the force on the cell boundary is a constant, a chemotaxis constant, lambda, times the gradient of the chemical concentration. And so then lambda chemo is the force on the cell membrane per unit chemical gradient. And in reality, that would be a rather complex function of the cell type, the density of receptors, the energy level of the cell, the cell's history, and so on. This kind of very simple chemical response model has some advantages. It's straightforward. Cells move up or down gradients depending on the sign of the lambda chemo. It does have some perverse effects. In reality, when cells are in a chemical gradient, if the gradient's too small, they don't move. As the gradient gets bigger, they move a little bit faster, but they have a relatively limited maximum speed. And so for strong gradients, the cell shouldn't move faster than for moderate gradients. With this kind of linear response, the cells move infinitely fast in very steep gradients, and they'll start breaking apart or disappearing, which is an artifact of the way we're doing the simulation. As I mentioned, our cell movement doesn't change the chemical field. In reality, when the cell moves, it should push the chemical out of the way. And in general, detecting a chemical field requires the cell to bind the chemical on its membrane. That removes that chemical from the surroundings. In some cases, the amount of chemical is large in the outside, and so the amount removed is negligible. But in other cases, you're following weak gradients, and in that case, you really should be removing the chemical when you detect it. The simplest possible thing to do computationally is to have a chemotactic effective energy. We haven't talked that much about how these effective energies work, because I've been trying to minimize the math, but we'll see this here, where we sum over the cells. We sum over the voxels inside of each cell, and we say that each voxel contributes an energy, which is lambda chemo, times the local concentration. If we do that, then the energy goes down when the concentration is higher, if lambda chemo is positive. The energy goes up if when the concentration is higher, if lambda chemo is negative. As we know, the algorithm that we've developed moves cells towards lower energies automatically. And so the effect of this is to create a force that's proportional to the gradient of the concentration. Yeah, Joel.
So in this case, a bigger cell would give us a better reading of the chemo. Okay. So if I'm doing something with chemotaxis, bigger cells would be a better application.
Well, I guess the question is what you mean by better. So one thing that you could do, and I think I thought we had a homework problem for it, Giuliano. Didn't you create a homework problem where you asked the question, in the same chemical field, with the same lambda chemo, as you change the size of the cell, how fast does the cell move?
I don't think.
If we didn't give that homework assignment, we should add that to the stack, or you've self-assigned yourself that homework assignment.
Okay. Yeah, I don't remember seeing that, but yeah, okay. What I mean by better is a finer reading or a better tuning or something like that, in the sense of having a direction better defined, since the cell has more voxels to analyze.
So it's certainly true that if you're trying to measure a gradient, it's easier to do that over a longer distance. That's essentially what the bacteria are doing. They can't measure it over their own size, so they move some distance, and they measure the change in time, which is the change in space. If you're big enough, you don't need to do that. If the chemical gradient on the outside is strong, steep, then you may not need to do it either. But certainly for weak gradients or in noisy situations, that's absolutely true. There are a couple of possible algorithms for determining the chemotaxis energy, and some of them have rather perverse effects. One question is, if you have two cells and you bring them in contact with each other, at the boundary between the two cells, is there any force? And this algorithm says, no, there isn't. Because if I replace a voxel with cell number 1 with a voxel with cell number 2, I still have a cell there, And the energy contributed by that is still the same. It's lambda times concentration. And so in this algorithm, the chemical field only acts at the boundaries between cells and medium. That may be right biologically, but there also are cells which respond over their surface, whether they're in contact with the medium or not. In that case, you have to use a different algorithm where the cell measures only the chemical difference when it's protruding and not when it's retracting. And in that case, you get a force on every boundary of the cell, not just on the boundaries that are in contact with the medium. And so there are a whole variety of choices here. If you look in the literature, people have, Pauline Hogeveg and her students were the first people to try to add this, which is why it's Glazier-Graner Hogeveg. And they explored a variety of these options, and we also explored a variety of them. Not all of the options people have tried are available in CompuCell. Several are, but not all of them. If we want to implement this computationally in CompuCell, then we use the Chemotaxis plugin. We have to say plugin name Chemotaxis. We have to say what chemical field we're responding to, and what the solver that it comes from is. Here, Diffusion Solver FE, name of the chemical field. And then we can define chemotaxis by cell type, name of the cell type, the strength of the chemical response, whether it responds on all surfaces or only some. Here, it only responds on contact with medium. And we can do that repeatedly. Again, we can have multiple specifications to sum up different responses. So the same cell could chemotax to multiple chemicals simultaneously. The same chemical field could attract different cell types in different ways. And so let's try doing that. Why don't we create a simulation, say 200 by 100 lattice, use periodic boundary conditions, X and Y, no flux in Z. Create a single cell, called cell if you like, add a chemical field, oxygen, using Diffusion Solver FE. And then specify lambda chemo of 500. And we want to create a linear gradient using initial conditions. Now here I said initial and boundary conditions, but I'd actually recommend turning the diffusion off, having diffusion constant, decay constant be 0, and just set up an initial condition. Put the cell initially on the right-hand side of the cell lattice, plot the position of the center of mass of the cell in the x direction versus time, and then run the simulation and change the value of lambda chemo and ask the question, how does the speed of the cell change as you change lambda chemo? I know, Connor, you and Andrew have probably done this a lot already. So you can either help the people who haven't done it before, or we can maybe explore some more complicated situations. But why don't people take a few minutes to try this out? If people have all done this one already, we can move forward with it. But let me give people the opportunity to. And for people who've already done this before, there are a number of things you could do to explore. One of which is looking at the different options for chemotaxis that CompuCell provides. Another one would be to sort of jump ahead and try to look at a question like, what is the speed as a function of lambda chemo? I haven't shown you how to change lambda chemo yet, though, so that means jumping ahead a fair amount.
So this is the exercise number 5, I suppose, where we have to change the chemotaxis lambda and compare with the velocity in
Mhm.
X. Now, my question is, I think I was expecting the cell to not disappear over time, since I think I'm reducing this as it goes towards the concentration, but in reality it goes there and sticks to this other cell and dies. We'll see over time.
This is a reasonably complicated simulation.
There's a lot of information here, right? Sorry, it's the way
So let's see. So what you've done here is you have multiple replicas of. I mean, this is like a little microfluidics experiment where you have how many? 10 channels?
11, yeah, 11 channels.
11 channels, and you're injecting chemical from one end in each channel.
Yeah, each one has a different lambda as a starter. The green one is, as you can see, just a point here is the last one. The brown one is the top one, and they have different. And then you start see them moving, and also you start see the points moving in that direction, which is the direction of more chemotaxis lambda. But there is a point that they go back, right? Because it's getting close to this more material. But they keep disappearing instead of getting there. The question is, I think there was a question number F, that is how to make it go back. And I wonder if there is like a negative, it's just a sign thing that we just need to change the sign to. Oh, okay. Okay, so maybe that's what I should do. Yeah, see, it goes back a little bit. It's the brown, you start seeing it going back after it disappeared now.
So, sorry, what are you? I'm trying to understand. Josh, go ahead.
Oh, sorry. I was going to say mine looks completely different. So I was concerned if I maybe had the wrong idea.
Show yours. Show yours.
Well, this isn't how I would have done it at all. Joel's done something very interesting, but not how I would have done it. So
Okay.
I think there's a lot of room for different opinions on this one. Let me go ahead. Let's see, Josh.
So mine, it has the, I don't know, it's the gradient is the same across all lanes, but the lambda is different.
Mm-hmm. Yeah.
And they do
so
have different speeds.
And so you started out with a predetermined gradient, right? You started out with the gradient already there.
Whereas
Joel started out with no chemical and started injecting it.
Okay.
So those are fundamentally different biological situations. And so the fact that you get a different result to me is not surprising, because in his case, initially there is no chemical and then you start introducing it. So the gradient's continually changing in time.
Mm-hmm.
Whereas in your case, you have a fixed gradient. So yours is getting a lot closer to what I expected to see, Josh.
Okay.
And it's not that Joel is wrong. He just chose to interpret the problem as a different biological situation. And here I see you've got the lambic reversing when the cell gets close to the end.
Yeah.
So Josh, Joel, this should answer your question, I think. So Josh has the lambda chemo change from positive to negative when you get close to the end. And you see the cell goes and then crawls and then reverses. So they sort of bounce off the ends, which is nice. And the one thing I'm not sure about is the velocity.
Yeah, I was confused about how I should go about that. Whether it just be calculating it from the center of mass x coordinate. Well, also I was just kind of confused because they asked about the y velocities too, and I don't know, it barely moves in the y direction, so it's very small. But
right, well, that was actually the point.
Yeah.
uh, if the chemotaxis is so the cells can move in these cases where the cells are in lanes, like here, they don't have the ability to move very much in the y direction, because even if they had a velocity, they'd bump into the wall. They couldn't move very far. But if we were in a free space, if we just had a single cell moving in an open space, then they could move in the y direction too, and that movement should be random. And so what you should see is that even if the instantaneous velocity is pretty big, you get a little bit of drift in y, but primarily what you see is movement in x. And so I would expect that the y velocity would essentially be near zero. It's not going to be absolutely zero, but near zero. Whereas the x velocity should depend on the value of the chemotaxis parameter. And so that looks fine. But what I'm not seeing here is that I would have expected that the velocity would basically be constant.
Yeah.
No.
I haven't been, it's always, I've tried a bunch of different iterations and it's always been really noisy.
Right. So the cheapest way to get the velocity in this problem is to measure the time at which do what the bacteria do, which is to measure your position and time at the start, and the position and time where you turn around, and say the velocity was the difference in position divided by the difference in time. And then do that repeatedly, because the instantaneous velocities are subject to a lot of fluctuation.
Okay.
But the velocity over the whole interval is well defined.
Okay. Yeah, I think this is only over 10 Monte Carlo steps. Yeah. So you're saying I should do it over 500?
Well, that would be 500 in space rather than in Monte Carlo steps.
Yeah. I mean, okay. Yeah.
So when you measure velocity, it's distance over time. And so you have a choice. You can say, I'm going to measure how far I went in 500 Monte Carlo steps. Or you can say, I'm going to measure how long it took me to go 500 voxels. And I'm suggesting that for this particular exercise, the second approach, which is measuring the time it takes to go from, say, position 50 to position 450, may be a more reliable estimator of the velocity than measuring the distance in a fixed time. Because the distance at a fixed time, if for one that's going fast, that's fine. If one's going very slowly, over 500 Monte Carlo steps, you might only move 10 voxels. And so then you'll still be noisy.
Yeah, yeah. It's just calculating the difference in position every 10 Monte Carlo steps.
Yeah.
So.
But if you simply up that to 500 to 100 Monte Carlo steps or 500 Monte Carlo steps, it probably looks smoother.
Okay.
And what I'm suggesting is basically just measure the velocity once for each direction. Average velocity over that, and that should give you a pretty well-behaved result. And then you should be able to see that as you increase lambda, the speed goes up.
Okay.
Great.
Thank you.
But that looks really nice. I mean, that's a different interpretation of the problem, but a very meaningful one. Great. Well, there's no point in then. I don't think there's any more point in spending time on that exercise since people have actually jumped way ahead of it. But that looks great. So just to remind you, contact, volume, flex, center, mass. I think you've seen all of this, so I don't need to go through it in detail. You've done it already. So maybe since we're almost out of time, did you do this exercise of self-constructed gradient?
I don't remember. Is this supposed to be the number 6? The only one that I haven't done yet is the number 6.
Okay, so let's do this quickly together. And you can use the code that you've written already. Or this is very quick to do using Wizard. So we've talked about the cell following a chemical field that's diffusing. And cells do that. But one problem with diffusion is that we know that the diffusion lengths for most of our biologically significant molecules are pretty short, like 100 microns. And that means that if you want to get a signal across a tissue that's centimeters across, you can't do it with diffusion. And in early embryonic development, in particular things like neural crest cell migration or lateral line cell migration, cells have to move long distances. In vascular development, cells have to move long distances. And the chemicals they're following have diffusion lengths that are short. And the question is, how do you get cells to actually move long distances following chemical gradients when the chemical gradients can't possibly be long enough to get those signals? And there's actually a sort of a paradoxical solution to that, which cells do use. And that's what's called a self-constructed gradient. So suppose that it's also the idea of digging your own hole. So suppose that I have a chemical that's not diffusing, that's bound to my extracellular environment, it's bound to my outside. And suppose that I eat it. This could be a bacterium eating nutrients, or it could be a cell taking a signaling molecule out of its environment. If I eat it, then where I'm sitting, the concentration is low, but ahead of me, the concentration is higher. And if I then move chemotactically up the gradient, I will move forward, and then I'll eat it there, and the gradient will move ahead of me, and I'll eat it again, and it'll move ahead of me, and I'll eat it again. And that will tend to cause me to move forward. Now, if the cell doesn't have an idea of a front and a back, and it can do this over any place on its surface, then it'll be like bacteria. I'll move forward, then I can reorient, move forward, reorient, and move forward. And so I'll get actually that kind of run and tumble movement that I'm talking about, that we talked about for bacteria. So what I'd ask you to do here in the last few minutes is create a simulation. I would just start with a constant of 100 by 100 with periodic boundary conditions. Define a single cell type in a single cell. Set the diffusion and decay constants globally to 0, but set a diffusion coefficient and a decay coefficient inside the cell, just for the cell. Set the initial concentration to be 1 everywhere, uniform. And you can make boundary conditions for the field to be periodic. Lambda chemotaxis 200. What happens? And that's really, really pretty easy. You can use, if you use the wizard, it takes about 2 minutes to set that one up. And it's a pretty neat result. So I'd like to end today with people trying that out. Is it clear what the assignment, what the little exercise is? And Giuliano, if you can code that, that would be great. So it's a backup. So I want to show people before this. I was trying to quickly rebuild it for myself just to keep up. I had player crash, so I have to. All right. So here's how I would do it. Plug in name chemotaxis. Here morphogen lambda chemo cell. And here I set the global diffusion constant, decay constant to 0, initial concentration of 1 everywhere, diffusion coefficient of 0.001 inside the cell, decay coefficient of 0.01. And here's what I got. Does anybody have something they want to show that's similar or different?
Mine is similar.
Okay, let's take a look.
It's moving around.
Yeah, that's good. Okay, so this is what I would expect to see. And you'll notice something interesting happens here. What happens when the cell tries to cross its original path?
It starts to turn back away from it, right? Because the concentration there is lower.
So it's doing what's called a self-avoiding random walk. It's moving, it's persistent because it tends to move in the same direction for some amount of time, but it's a random walk because if you wait long enough, the direction changes. But it's self-avoiding because it can't cross. And so what you'll see is that if you imagine a bacterium eating in its environment, it will gradually eat all of the chemical that it can find. If it really runs out completely, what will happen is it'll move randomly because there's no information anymore in the environment. So it's not an absolute rule that it can't cross itself. Let's see what happens now. It sort of locked itself in here, right? So what's it going to do?
Well, this is right now. This is different,
So
though, right? Because there's still.
So it crossed.
relative.
It did cross its back. It did cross its old path. It didn't want to do it, probably. It preferentially finds the high concentration. But if it really gets stuck, then it'll cross its own path. So it's not a perfect self-avoiding random walk. And so with this, you can play a lot of games. When you change the size of the cell, it changes the effect. When you change the diffusion constant inside the cell, it changes the effect. When you change the number of cells, you can have cells compete. And if you have a bunch of cells, they'll tend to align with each other because they'll repel each other effectively and move in a coherent way through the environment, eating the food, if you imagine it that way. The other thing that one can do with this, can be quite interesting, and those were the extensions that I had in my slide, but we won't get to tonight, is that you can ask the question, in many biological situations, either the diffusion constant outside isn't completely zero, so there's a little bit of diffusion outside, in which case these gradients will gradually smear out over time. And so the cell will forget that it was in a given region, or rather the region will forget the cell was there. And the other thing that can happen is that the environment can regenerate the chemical that's been destroyed. And again, there's a forgetting that happens. And the time scale of that forgetting versus the time scale the cell takes to eat all of the chemical creates a competition. If the forgetting is fast, the cell will move will not self-avoid. If the forgetting is slow, the cell will do this kind of self-avoiding random walk. And so you can actually generate really a whole bunch of different trajectories from this. And you can do something like plot just the center of mass position of the cell. It's a little bit hard here to see what the trajectory really was. There are ways then of analyzing what's called the persistence length. Just how far does the cell go typically in a given direction before it changes direction, using a correlation function. That's a little bit more work to calculate. And then you can see how that correlation length depends on these parameters. And this is, as I mentioned, a very efficient way for, say, bacteria to eat all the food in their environment. You'll notice that by the time you haven't been talking that long, and it's eaten almost all the red in its environment. It's also a way that you can get persistent movement down long distances, which is used in pathfinding, in neural pathfinding, in neural crest cell migration, in epithelial, vascular endothelial cell pathfinding. And it also, with a few modifications, can be used to lay down a track where you have a leader cell that moves through, and it sets up a track of gradients that then allow follower cells to follow in the same direction. That can be set up as well. And so I wanted everybody to see that because for me, it's a very simple simulation. It didn't take you very long to build it. And it's actually got a lot of very profound biophysics and biology in it. So I'd like to, maybe that's the place to end for today. I'll just say that if you're interested, I had thought of some exercises, as I say. One would be to write a little extension to plot the center of mass position of the cell. And then if you're really ambitious, you could look up what the definition of persistence length is and see how you can calculate it from the trajectory. That's work. That's real work. It's not terrible, but it's real work. Okay. So that's where we'll stop for today. Any questions before we break? I think I'll leave the homework assignment up. Maybe Giuliano, you could generate a homework problem around this self-constructing gradient to add for this week. But I know people were saying they weren't finished with the homework, so I hope this helps a little bit with that. And if there aren't any more questions, I'll say good night.